Good afternoon, everybody. Today we're going to be jumping into chapter 12. This looks at basically the central dogma of biology, how we get from DNA and how we ultimately get with this end product called a protein. So that's what this chapter basically looks at. There's a lot of little intermediate steps and there's a lot of words on some of these slides here, but you'll be okay. I'll highlight some important aspects, some of the major bullet points, and hopefully you'll be able to recall some of those for the exam. We would be remiss if we didn't go back and look at some of the history, just a little bit of it, to kind of understand where we are today. Because right now, when we talk about, passing genetic components on to the next generation, it seems pretty obvious. It's DNA. Mom and dad's DNA hook up and mix together and give us the beautiful thing that we see in the mirror every single day. But it wasn't always like that. There was a vast number of scientists who didn't understand it, didn't believe in it, called it something else. And we hinted at some of that when we talked about Gregor Mendel. He just referred to them as factors. He didn't really understand kind of what they are, how they even went from one generation to the next, because some of his stuff or some of the traits that he was observing, they weren't all sex related. They were, there are some traits that are passed on via the sex cells, and there are some that are not passed on by sex cells, autosomal and the sex cells. And he didn't really understand any of that. So this concept of DNA was very foreign, and even evolutionary theory, when it was first postulated, they didn't really understand the genetic component of it. That was not yet fully understood. So it's something that we take for granted now, but it started way back in history, right? As they say. They were trying to figure out what was the transformative factor. Frederick Griffin, he looked at the virulence of Streptococcus, specifically Strep pneumoniae. He was concluding, coming with evidence, that the virulence could be passed from one dead strain to a non-virulent living strain, and he called it, transformation. People after him, after Frederick Griffin, Avery, and a number of other ones, and et al. Just means others. They actually found out that it was not necessarily the virulence that was being transmitted from one organism to another, in this case from one strep to another strep, but it was actually the DNA from the dead cells that was being incorporated into the genome of the living cell. So Frederick Griffin came up with this concept, but he had no idea what in the hell was even going on. So he just referred to it as transformation. It was transforming. It was transforming this one thing into something that wasn't virulent into something that was pretty deadly. So, but he didn't understand it was the DNA that was giving the blueprint for how to do that. Avery and a number of other scientists here, and I have some of them listed on the next slide. They're actually the ones that discovered this transformative factor, if you will, was the DNA. They looked at DNA, they looked at proteins. There was a camp that said, well, it's all protein related. There was a camp that said it's this stuff called DNA. And so they had to go through and do studies to try to isolate all the various different proteins and see if that was a transformative factor or if it was truly this DNA, which ultimately we know now gives rise to the proteins, right? So they looked at it. They had to have some requirements. They came up, must be able to store information. And we really, at this time, didn't really fully understand some of the concepts of, and this is early 1800s going into the early 1900s, didn't really know that DNA could store all this vast amount of information that we do know now. Had to be stable enough. We couldn't stabilize it necessarily in the beginning to fully grasp that. That didn't come till a little bit later in the 1900s. And so being able to really kind of see it, see how it mutates or how it undergoes mutations and able to confer that onto the next generation. Um, until Avery and the colleagues of Avery started doing some experiments with DNA and proteins, they didn't really know about that until after all of these studies. So again, this is kind of what I'm kind of talking about here, and Avery and et al. Avery, McLeod, and McCarty's all of their experiments. They took DNA and activated it, with an enzyme, broke it up, did the same thing with RNA, did the same thing with proteins. Okay, DNA. And anytime you see ASE on the end of something, it's an enzyme that breaks it down. So they looked at Streptococcus and they injected all of these various different enzymes and then to see what this quote unquote transformative factor was. And it showed that DNA was the substance that was actually passing on this virulence, because once they applied that, nothing happened. When they applied the RNA, stuff happened. When they applied the proteases, stuff happened. So we destroyed the RNA, they destroyed the proteins, and the only one that it was still able to have change in subsequent generations was the one with the DNase. So the DNase destroyed the DNA and therefore destroyed the blueprint for how this thing can be really virulent. So that's how they figured it out. So now they're trying to figure out here with Hershey and Chase, now they're trying to figure out how. Now they're trying to see it actually happening, okay? Avery and McLeod and McCarty's experiments, they proved that DNA is the biz, is the big dog, okay? So now we want to see how that DNA gets from one individual to another, okay? Because we have these things called viruses, and viruses can inject their DNA into a host cell and get the host cell to do its bidding, right? So now they're like, okay, so let's see if we can watch this transformative factor, this DNA, and let's see if we can actually visualize it and see it going from one individual cell, like a virus, into another individual cell. So they use this thing, Henry and Chase's experiment, they use this thing called a T phage. It's just a virus, okay? They gave it a radioactive label. That way they were able to follow it via various imaging studies, okay? So they discovered that the radioactive labeled DNA, but not the protein, was the stuff actually getting into the bacterial cell and causing the bacterial cell to make what the virus wanted it to make. And that was the transformative factor. So even though they understood that it was DNA, they still wanted to see it, okay? And again, they did a study with radioactive DNA, radioactive everything. And the only thing that was shown to get passed from one to the next was the DNA. And here you can see the virus on the outside. This is the bacteria. And here it's injecting its DNA inside this this poor little bacterial cell. And then this bacterial cell is going to eventually incorporate that DNA into its own. Remember, it's circular in nature. And it's going to do whatever the virus's DNA told it to do, okay? Make more viral components.
Material. The virus consisted of a DNA molecule surrounded by a capsid made of protein. When viruses infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, viruses were grown in a medium containing radioactively labeled amino acids. This resulted in a virus population with labeled proteins, but DNA that was not labeled. The viruses were then allowed to infect the bacteria. The viruses attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The viruses produced in the bacterial cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with the production of new viruses in the cell. In the second part of the experiment, viruses were grown in a medium containing radioactively labeled deoxyribonucleotides, the building blocks of DNA. Viruses were produced with labeled DNA, but unlabeled proteins. When the viruses infected the bacteria, the radioactively labeled DNA entered the cell and could be found in viruses produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of viruses.
So today, now we know that all of these transformative factors, it's all the DNA. That's what it is. And for us today to understand that, I mean, this is a valuable tool, and this is what we're going to talk about in chapter 14. By understanding what that transformative factor is, the DNA, well, good golly, Miss Molly, I can take this DNA and I can harvest it from whatever I want. And I can, through fancy pants science, get this into a virus and then get this into a host cell where I want that specific outcome. Like, do I want stuff to glow? I can take the glowing aspect of a jellyfish and I can inject that into other cells, other organisms, and I can get certain things to glow. I can get things to glow that I want to tag. If I want to figure out how a arm manifests in utero, I can tag what I know happens to be the genes associated with making arms and legs, and I can tag it in the DNA, and I can tag it and I can visualize it. And these are great, fantastic things. They've done it for other various organisms as well to see if they can get the DNA from one host animal into another animal. And they get these other organisms to kind of glow in the dark. So there's a lot of potential uses. Having a glowing rat or mouse probably isn't, earth shattering, going to change the world. But it did show that DNA was the factor. And if I can isolate the specific genetic information that I want, I can get that spliced into another organism and I can get that organism to exhibit it. So that has some very cool ramifications and some very scary ramifications as well. So when we look at the genetic material, when we look at DNA, there are 4 bases, right? Adenine, guanine, cytosine, and thymine. Those are our 4 DNA amino acids, okay? Adenine, guanine, thymine, cytosine. They're purine and pyrimidine bases. I don't really expect you to remember that. I got a bunch of other fun stuff to remember, but these are the 4 nucleotides associated with DNA: adenine, guanine, cytosine, thymine. A dude came along named Shargoff, and he came up with some rules associated with this. He did a bunch of research into DNA, looking at DNA from a multitude of number of species, and he came up with these rules. The amounts of A, T, G, and C in DNA, in a DNA molecule, are the same for every member of a species. Seems pretty obvious, right? Every Homo sapien has the same amount of A, T, Gs, and Cs in their DNA. It varies from species to species, but within that same species, it has the same DNA, which makes sense because, we want these things to procreate. We want the DNA to jive, and we want them to be able to have viable offspring. So to me, that makes some sense. But for Shargoff, this is what he says. Adenine is always equal to thymine, and guanine is always equal to cytosine. Okay. This suggests that there is complementary base pairing. So when DNA gets copied, okay, when we separate the DNA molecule out, okay, remember it's a double helix, we straighten it out, open it up, and make a copy. A always goes with T, G always goes with C. Always, always, always. Okay, always, always, always. That we maintain the integrity of the DNA molecule. So whether it's dividing through mitosis or through meiosis, I always get the same thing. Unless there's a defect. You know, I get it. We talked about those. But I'm always getting A with a T. I never get A with a C. I never get A with a G. And it has to do with its structure, okay? The chemistry of its structure, which we're not going to really get into. And you can see there's some numbers there. Just give them to, visualize. I mean, this is a hell of a lot of stuff. Each chromosome has on average about 140 million base pairs. Whew, damn, that's cray. And the number of possible nucleotide sequences is 4 to the 140 millionth power. That is a massive, massive, massive number. Can't even comprehend that thing. Here's what the DNA, here's what these nucleotides look like. Okay. Again, I probably won't ask them on an exam. I'm not going to ask you to differentiate these from a picture or anything like that. It's just to kind of see. Okay. Adenine always goes with thymine. You can see they're pretty similar in structures. Guanine always goes with cytosine. Pretty similar in structures. Okay. So we have visualized, we figured out that this transformative factor is DNA, not proteins, not necessarily RNA. We have seen the DNA actually going inside another cell and injecting into it and making it do its bidding. So now we want to see what that actual genetic material, chromosome, looks like. Enter in Miss Rosalind Franklin. She studied the structure of DNA, but she was studying the structure of DNA using X-rays. And she found out that if you concentrate it enough, so get enough genetic material in there, you can get it to become this viscous solution. And you can separate its fibers out. And under the right conditions, you can even get an image of it. And here's kind of this diffraction, okay? And there's your, there she blows. There's your X, okay? Diffraction is just using a beam of light and the different wavelengths that go through it with the various part of the light spectrum there, allows it to pass through certain aspects of it and not through certain aspects of it, and it gives you that appearance. So that's what that is. But she produced a series of all of these diffractions, these X-ray diffractions, and she was able to prove that DNA was double-stranded, DNA was a helix, and because of the staining technique, that some of it was repeated. Enter in Watson and Crick. Some of their contemporaries, some of their colleagues, as you can kind of see there, showed Mr. Watson, Dr. Watson, what Miss Rosalind Franklin was doing. And then Watson and Crick took it and then ran with it and came up with the model of DNA and got a Pulitzer Prize out of it, and right, leaving everybody else in the dust. Okay, and this is in 1953. Okay, Dad was born like 1950, so good God, that's crazy. That's not too long ago. Came up with this double helix model. Okay, and it twists around on itself. We have the sugar phosphate backbone making up the sides, deoxyribose. And remember those things, hydrogen bonds? There we go. That makes up the rungs. If I go forward here, here's my sugar backbone right there. sugar backbone. There's my hydrogen bonds. And there's my A's with T's, G's with C's. Always, always, always. They eventually kind of came up with this model with everybody else's help. And then they also figured out that DNA has two ends to it, a 5' end and a 3' end. And this is important for basically figuring out how this thing is copied and how this thing is read. It's always read in the 5' to 3' direction, okay? It's always read in that way. So it gets copied very easily in that direction, okay? The other one, the other segment of DNA will be opposite to it. And it will actually be, so if this is 5' and this is the 3' end, the complementary base pair will be opposite 5', 3'. Okay, so it gets easily read in this direction here, and this one not as easy, but we'll kind of see what that looks like here. And as you can see, they kind of won the Nobel Peace Prize in science there in 1962, and just Jeopardy knowledge for you to use at a party, and you can impress somebody.
The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine. The backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands.